Let's look at question number 12 from GATE Biotechnology 2018 paper. So the question is, which one of the following techniques can be used to study conformational changes in myoglobin? So option P, mass spectrometry, fluorescence spectrometry, circular dichroism spectroscopy, and light microscopy. So the options are P only, and B, P and S, and option C, Q and R, and option D, S only. So let's learn about these different techniques to be able to answer the question. So spectroscopy is the study of the interaction between matter and electromagnetic radiation. So historically, spectroscopy originated through the study of visible light dispersed according to its wavelength by a prism. The electromagnetic spectrum is the range of frequencies of electromagnetic radiation and their respective wavelengths and photon energies. So the term spectroscopy defines a large number of techniques that use radiation to obtain information on the structure and properties of matter. So the basic principle shared by all spectroscopic techniques is to shine a beam of electromagnetic radiation onto a sample and observe how it responds to such a stimulus. So the history of spectroscopy began with Isaac Newton's optics experiments. So Newton applied the word spectrum to describe the rainbow of colors that combine to form white light and that are revealed when the white light is passed through the prism. So during the early 1800s, Joseph von Fraunhofer made experimental advances with dispersive spectrometers that enabled spectroscopy to become a more precise and quantitative scientific technique. So, spectrometer is an apparatus to measure spectrum and show intensity as a function of wavelength, frequency, energy, or mass. Let's now learn about mass spectroscopy. So, mass spectroscopy is one of the primary spectroscopic methods for molecular analysis available to an organic chemist. So, it's a microanalytical technique requiring only a few nanomoles of the sample to obtain characteristic information related to the structure and molecular weight of the analyte. So, it is not concerned with non destructive interactions between molecules and electromagnetic radiation. Mass spectroscopy is the most accurate method for determining the molecular mass of the compound and its elemental composition, just as the name suggests. So, in this technique, molecules are bombarded with a beam of energetic electrons, and the molecules are ionized and broken up into fragments, and some of them are positive ions. So, each kind of ion has a particular ratio of mass to charge, that is m by e ratio, and for most ions the charge is 1, and thus the m by e ratio is simply the molecular mass of the ion. Mass spectra is used in two general ways. So one is to prove the identity of two compounds, and two is to establish the structure of a new compound. So the mass spectrum of a compound helps to establish the structure of a new compound in many different ways. So it can give the exact molecular mass, and it could give a molecular formula, or it can even reveal the presence of certain structural units in a molecule. So the basic theory behind mass spectroscopy is that it deals with the examination of the characteristic fragment ions arising from the breakdown of organic molecules. So a mass spectrum is the plot of relative abundance of ions against their mass to charge ratio. So the basic aspect of organic mass spectroscopy consists of bombarding the vapor of an organic compound with a beam of electron accelerated from a filament to an energy of 70 eV to form positively charged ions. The additional energy of the electrons is dissipated in breaking the bonds in the molecular ion, which undergoes fragmentation to yield many neutral or positively charged species. So this fragmentation may result in the formation of an even electron ion and radical. So the various positive ions formed can be accelerated and deflected by electric or magnetic fields. So the deflection of these ions depends on the mass, charge, and the velocity. So for a given charge, velocity and deflecting force, the deflection is less for a heavier particle as compared to a lighter particle. So thus a number of beams each containing ion with the same m by z values are obtained, and the beams are then made to strike against a photographic plate where they don't not only appear as separate lines, but the intensity of each peak as a result of the bombardment is recorded. So the clear visual presentation of a mass spectrum is usually obtained by plotting m by z values against the relative abundance, assigning the most abundant ion in the spectrum as 100%. So that is the base peak. So, although organic mass spectrometry is routinely used along with infrared, NMR, and UV for structural determination, its basic theory is different from the rest of these techniques. So, in mass spectrometry, no characteristic selective absorption of radiation is involved, as in the case of these other three methods. And then in mass spectrometry, the compound undergoes irreversible chemical changes, unlike the others, where the changes are reversible physical changes. Like once the sample has gone through a mass spec, the sample cannot be reused. So the mass spectral reactions are much more drastic than usual chemical reactions. It involves the production and separation of ionized molecules and their ionic decomposition product, and finally the measurement of relative abundance of different ions produced. It is thus a destructive technique in that the sample is consumed during the analysis. So, in most cases, the nascent molecular ion of the analyte produced fragment ions by cleavage of the bond, and the resulting fragmentation pattern constitutes the mass spectrum. So, thus, the mass spectrum of each compound is unique and can be used as a chemical fingerprint to characterize the sample. So here is a diagrammatic representation of how the mass spectrometer in instrument looks like. So and several steps of the whole process takes place in each of these different sections of the instrument. And so let's look at each of these methods, like processes: ionization, acceleration, deflection, and detection, in the next few slides. So the first step is ionization. So the atom is ionized by knocking one or more electrons off to give a positive ion. So mass spectrometers always work only with positive ions, and the positive ions formed will always carry a positive charge of +1. So these positive ions are persuaded out into the rest of the machine by the ion repeller, which is a metal plate carrying a slight positive charge. So the vaporized sample is passed through this inlet, and it is radiated with a beam of electrons over here, and the positive ions are emitted, and the positive ions, they do not go this way because of the ion repeller, which is also positively charged. So because like charges, they repel each other, the positive charges are like repulsed to the opposite direction, and hence the ions, they move out of this chamber and they move to the next part of the instrument. The next step is acceleration. So the positive ions now, they are accelerated so that they all have the same kinetic energy. So the ionization chamber, it has a voltage of like 10,000 volts, and the ion beam passes through this chamber. So the positive ions are repelled away from the positive ionization chamber, and they pass through three slits with voltage in decreasing order. So it starts with 10,000 volts and the voltage keeps decreasing, and at this third slit the voltage is 0 volts. So the middle slit carries some intermediate voltage. So all the ions are accelerated into a finely focused beam of ions. The next step is deflection. So the ions are then deflected by a magnetic field according to their masses. So the lighter particles are deflected more and the heavier particles are deflected less. So the amount of deflection also depends on the number of positive charges on the ion. So the more positive ions the ion has, the more it gets deflected. So like we just saw, the deflection of the ions depends on the mass of the ions and the charge of the ions. The next step is detection. So the beam of ions passing through this machine is detected electrically. So when the ions hit the metal box, the charge is neutralized by an electron jumping from the metal on to the ion. So as you can see here, the ion stream that is passed through the deflector, it makes through the detector machine. It makes through this the ion detector chamber. And here what happens is, so here's a positive ion, and other ions collide with the walls where they will pick up electrons, and hence the positively charged ion gets neutralized because an electron is knocked into it. So they get removed from the mass spectrometer by the vacuum pump. So this process, the neutralization of the positively charged ions, it leaves a space among the electrons in the metal and the electrons in the wire shuffling along to fill it. So a flow of electrons in the wire is detected as an electric current, which is amplified and recorded, and the more ions arriving, the greater the current is. So one of the processes that is involved in mass spec is fragmentation. So the bombardment of molecules by the electron beam with energy between 10 to 15 eV usually results in the ionization of molecules by removal of one electron. That is the formation of molecular ions. So this is the first step of the whole procedure, which is ionization. So when the energy of electron beam is increased between 50 to 70 eV, these molecular ions acquire a high excitation, resulting in their breakdown into various fragments. So this process is called fragmentation. So as a result of fragmentation, several types of ions are formed. So they are molecular ion or parent ion, which forms the big peak in the mass spec reading, and then fragment ions, which result in smaller peaks, and also rearrangement ions, multi-charge ions, negative ions, and metastable ions. So by reading the peaks, we could know what molecule is present in the sample. So here are some general rules for interpreting the mass spectrum. So in order to interpret the mass spectrum, one should attain an understanding of the ionization process, and you should know how to observe the fragmentation pattern. So the exact molecular weight is the molecular weight of a pure compound from the identification of the parent peak. So the molecular weight one can determine molecular formula. And and then there's isotope effects. So heavy isotopes will exhibit peaks in a mass spectrum at m by z ratio, one or more units higher than the normal. So that indicates that there are isotopes of molecules present. So there will be small peaks at m plus 1 and m plus 2. For example, like for nitrogen, the molecular weight is 14. But if we see a peak at 15, it means that an isotope of nitrogen is present. So here's a simple readout from the mass spec. And so the x-axis denotes the mass by charge, m by z values. So this format is used because a m by z ratio of 28 can represent a mass of 28 with a single charge, or it might be 56 with a charge of 2, because 56 divided by 2 is 28. So it could just indicate like two molecules of the molecular weight 28. So a single charge is the most common, so the x-axis best equates to mass. And then y-axis indicates abundance. So in a standard mass spectrum, the uh, the y-axis is made relative to the largest line, which is the base peak. So it will run from 0 to 100%. So the relative abundance would be 0 to 100%. So absolute abundances are typically used in GCMS spectra, which is like gas chromatography and mass spectroscopy. So they will run from 0 to the highest recorded line intensity. So those are absolute abundances, whereas in relative abundances, everything is normalized to the highest base peak. So what are the types of information you can get from this graph? So you could get information about the mass of the fragments and the presence of isotope lines and gaps between these lines. So let's look at some of the basic information. So here's a spectrum for air, which is like a mixture of nitrogen, carbon dioxide, and oxygen, but predominantly nitrogen and oxygen. So if you look at this peak, it's 28. So it would be nitrogen N2, which is 14 plus 14, 28. And also 29 could indicate N15, which is an isotope of nitrogen N14. So it is small because N15 is not very common in nature, because that is why you could see very less abundance of the isotope N15, but highest abundance for like normal nitrogen N14. So we know that most of the small lines could be because of the isotopes, but what could 16 and 17 be from? So these fragments could indicate water, and water shows fragmentation where it can lose one or two hydrogen atoms. So you'll see that the fragmentation patterns are very important for mass spectroscopy. So 18 would indicate water, H2O, because 16 plus 1 plus 1, H2O gives a mass of 18. So when you see 17, it could be OH, a hydroxyl group, where one hydrogen atom is lost, and 16 could indicate simply an oxygen molecule. Also, a peak up to 100% abundance is called a base peak, and M is a parent ion peak, which is usually the next largest peak to the base peak. It is also called molecular ion peak. So molecular peak of M produced when an electron ejected from the molecule is M plus 2 electrons, a protonated M plus 2 electrons. And here are some of the exact masses and natural isotopic abundance and some ratios. So these are some values you could remember, like to easily interpret what mass indicates what molecule from a mass spectrum reading. One of the techniques is fluorescence spectroscopy. So, fluorescence is the emission of light by a substance that has absorbed light or other electromagnetic radiation. So, fluorescence spectroscopy or fluorimetry or spectrofluorometry is a type of electromagnetic spectroscopy which analyzes fluorescence from a sample. It involves the use of a beam of light, usually UV light, that excites the electrons in molecules of certain compounds and causes them to emit light of a lower energy, typically light in the visible range. So, a complementary technique is absorption spectroscopy. So, devices that measure fluorescence are called fluorometers or fluorimeters. Fluorescence spectroscopy is a fast, simple, and inexpensive method to determine the concentration of an analyte in solution based on its fluorescent properties. So, it can be used for relatively simple analysis where the type of compound to be analyzed is known, to do a quantitative analysis to determine the concentration of the analytes. So, fluorescence is used mainly for measuring compounds in solutions. So, in fluorescence spectroscopy, a beam with a wavelength varying between 180 and 800 nanometers passes through a solution in a cuvette. So, we then measure from an angle the light emitted by the sample. So, in fluorescence spectrometry, both an excitation spectrum, the light that is absorbed by the sample, and or an emission spectrum, the light emitted by the sample, can be measured. The concentration of the analyte is directly proportional with the intensity of emission. There are several parameters influencing the intensity and shape of the spectra. So, when recording an emission spectrum, the intensity is dependent on the excitation wavelength, concentration of the analyte solvent, path length of the cuvette, and self absorption of the sample. There are different kinds of instrumentation. So, they could be filter fluorometers or spectrofluorometers. So, filter fluorometers use filters to isolate the incident light and fluorescent light, and spectrofluorometers use diffraction grating monochromators to isolate the incident light and the fluorescent light. So these are the basic components of a spectrophlorimeter. The light from an excitation source passes through a filter or a monochromator, the first monochromator, and passes through the sample. Here, some of the light is absorbed, making some of the molecules in the samples to fluoresce. So, a part of the fluorescent light is then focused on a filter or a monochromator to the second monochromator, which is often placed at a 90-degree angle to the excitation light. The light is then detected by a detection device, and various light sources may be used as excitation sources, such as lasers, photodiodes, xenon arcs, and mercury vapor lamps. A laser only emits light of high irradiance at a very narrow wavelength interval, typically under 0.01 nanometers, which makes the excitation monochromator or filter unnecessary. So the disadvantage is that wavelength of a laser cannot be changed much. So a mercury vapor lamp is a line lamp, meaning it emits light near peak wavelengths, contrary to the xenon arc, which have a continuous emission spectrum with intensity in the range 300 to 800 nanometers. Filters, which are the monochromators, may be used in fluorimeters. A monochromator transmits light of an adjustable wavelength with an adjustable tolerance. So the monochromator can then select which wavelength to transmit. So for following anisotropy measurements, the addition of two polarization filters are necessary, one after the excitation monochromator or filter and one before the emission monochromator or filter. So no monochromator is perfect, and it will transmit some stray light, that is light with other wavelengths than the targeted wavelength. So an ideal monochromator would only transmit light in the specified range and have a high wavelength independent transmission. And then the detector can either be single channeled or multi channeled. So the single channel detector can only detect the intensity of one wavelength at a time, while the multi channel detector detects intensity of all wavelengths simultaneously, making the emission monochromator or filter unnecessary. So when measuring fluorescence spectra, the wavelength of the excitation light is kept constant, preferably at a wavelength of high absorption, and the emission monochromator scans the spectrum. For measuring excitation spectra, the wavelength passing through the emission filter or monochromator is kept constant, and the excitation monochromator is scanned. An excitation spectrum, which is also called the absorption spectrum, and an emission spectrum of a fluorescent compound can be made. The absorption spectrum tells us which incoming wavelengths are absorbed by the solution. With the emission or fluorescent spectrum, we can see which wavelengths are emitted after absorbing the incoming light. So these fluorescence spectrometers are generally used in biochemical, medical, and chemical research fields for analyzing organic compounds. So these are used in differentiating malignant skin tumors from benign. In analytical chemistry, the fluorescent detectors are used with chromatography, like high performance liquid chromatography. Circular dichroism spectroscopy. The next technique is circular dichroism spectroscopy. So, it is a form of light absorption spectroscopy which is used in determining molecular behavior in a broad spectrum of perturbation by measuring the difference in absorbance of light circularly polarized and left circularly polarized light. So, light could be of different polarizations. So, unpolarized light is where the direction of oscillations randomly changes in a same plane with time. And plane polarized light is when magnetic and electric field components oscillate in a definite plane being perpendicular to each other. And circularly polarized light is when magnetic field remains oscillating but electric field vector changes direction in a rotatory circular motion. So, in simple terms, circularly polarized light is where the electric field is rotating and the magnetic field is oscillating left and right like this, denoted by the arrow marks. And the direction of the propagation of the light is this way. So this represents a circularly polarized light beam. And these shows the other form of polarized light. So linearly polarized light, where the electric and magnetic fields oscillate and perpendicular to each other, and unpolarized light is where oscillation happens in random directions. Also, so where does this polarized light come from? So few golden scrab beetle like Chrysnia gloriosa and a few mantis shrimp have been found to generate circularly polarized light. And further, the circularly polarized light can be categorized as right hand circularly polarized, which is clockwise and abbreviated as RCP, and left hand circularly polarized, which is anti-clockwise. Let's now look at the mechanism of action of circular dichroism spectroscopy. So, electric field of a light beam causes a linear displacement of charge when it interacts with molecules. So, the magnetic field of the light beam, on the other hand, it causes circulation of charge. So, these two motions combine to result an excitation of electrons in a helical fashion. So, but the radiation used being chiral, it interacts in different fashion with a definitely chiral entity. So, after interaction, the electric field vector traces out an elliptical path. So, circularly polarized light becomes elliptical polarized light. So, here's a diagrammatic representation. So, light sources like short arc xenon lamps, they emit the light, and white unpolarized light is passed through the monochromator and it gets linearly polarized. So the linearly polarized light then passes through the photoelastic modulator, and then this gives circular polarized light, both RCP and LCP, and this then passes through the chiral specimen which is under the experiment, and then finally it passes through the detector. So what actually happens here? So when passing through an optical active medium, the speed between the RCP and LCP light changes along with the change in their wavelengths and their absorptivity. So now wavelength dependent difference of absorption is measured and plotted to get the circular dichroism spectra of the sample. So CD is measured by the the difference in between the absorptivity of LCP and RCP. So it's delta epsilon. So in some cases, apart from delta epsilon, which is the change in absorption between the RCP and LCP, molecular ellipticity theta is also measured. The relationship between delta epsilon and theta is given by this equation, where ER and EL are the magnitudes of the electric field vectors of the RCP and LCP respectively. So now what exactly do these values mean, and how does it tell anything about the structure of the molecule or the protein? So the circular dichroism signal arises from differential like RCP and LCP due to the chiral nature of the biomolecules. So the CD spectrum typically consists of two main features. So one is the negative band and the other is the positive band. So the negative band, the N band, is where the absorbance of left-handed circularly polarized light is higher than the right-handed circularly polarized light. It is often associated with a decrease in molecular ellipticity and is characteristic of certain secondary structures. So when like the delta epsilon value is negative, it means that the molecule is less elliptical, like it's not very elliptical. And a positive band is where the absorbance of right-handed circularly polarized RCP is higher than the LCP. So it is associated with an increase in molar ellipticity and is also characteristic of specific secondary structures. So the shape and the position of these bands in the CD spectrum are indicative of the types and amounts of secondary structure present in the sample. So common secondary structures in proteins are usually alpha helices and beta sheets and random coils, like you all would know. And each type of secondary structure contributes differently to the CD spectrum allowing researchers to analyze and interpret the secondary structure of the sample. So this shows the spectrum of light, and so vacuum UV spectrum ranges between 100 to 200 nanometers, and near UV ranges from 250 to 350 nanometers, and near infrared 750 to 950 nanometers, and far UV 200 to 250, and the UV visible spectrum, it's the range of all the visible colors you can see along with the UV, it's 350 to 750 nanometers. So, like, to give a more detailed explanation, far UV circular dichroism, which is 200 to 250 nanometer, is used to investigate secondary structure of proteins. Change in protein secondary structure as a function of temperature and concentration can be known, which allows us to know the enthalpy and Gibbs free energy of the process. And then near UV CD spectroscopy, 250 to 350 nanometer range. So this spectra provides information on the tertiary structure of proteins. It provides information on the nature of the prosthetic group present in the protein. And UV visible spectrum ranging between 350 to 750 nanometers in the visible region is found only when metal is in chiral environment, and free metal ions in solution are not detected by this visible range, and this can be used to detect protein bound metals. And further, near infrared CD, with the range of the spectrum between 750 to 950 nanometer, can be used to investigate geometric and electronic structure by probing metal DD transitions. Circular dichroism spectroscopy has several applications. So it's used to know whether the protein is folded or not, and if so, then the characterization of its secondary and tertiary structure and its structural family is identified. And then it's used to compare the structure of a protein obtained from different sources. And also, it's used to study conformational stability of a protein under perturbation of environment caused by heat, pH, denaturants, stabilizing agents, and so on. So it just helps to study the how stable the protein conformation is. And it's also used to study the effect of protein-protein or ligand-protein interactions. The advantages of this technique is that minimum quantity of sample is required, and it does not require extensive data processing, and it can be effectively used in the presence of multiple cofactors. It also has several complications and disadvantages. So, carbohydrates cannot be easily studied in this method, and aqueous buffer system used in circular dichroism spectroscopy often absorb in the range where structural features exist, and the system should be completely devoid of oxygen to perform the experiment below 200 nanometer wavelength of the light, and phosphate, sulfur, carbonate, and acetate buffers cannot be used until they are prepared in the range of 10 to 40 millimolars, and a large number of organic solvents cannot be used. And instead of ordinary xenon arc lamps, the use of high pressure short arc xenon lamps are essential for doing low UV CD spectroscopy. Let's look about light microscopy. The next given option was light microscopy. So before that, a microscope is an instrument used to see objects that are too small to be seen by the naked eye. So microscopy is the science of investigating small objects and structures using such an instrument. So there are two types of microscopes. They are light microscopes and electron microscopes. Let's have a brief introduction about light microscopes. So in 1590, Janssen and Zacharias Janssen constructed the first simple light microscope. So when molecular detail is not required, the light microscope is ideal and hence essential instrument for a biologist. So like when you want to know the morphology or something, the light microscope is good, but you can't know about the molecular details. So the simplest form of light microscope consists of a single glass lens in combination. So all modern light microscopes are made up of more than one glanceless lens in combination. The light microscope operates on the principle that light energy will pass through and around a thin object such as a microorganism, and with the aid of lenses, it forms a magnified impression on the visual sensory layer of the eye. The main components of the compound microscope include a light source that is focused at the specimen by a condenser lens. The source of light is usually the sun or ambient indoor light. Light microscopes are compound microscopes, which means that they contain at least two lenses. So the lenses are curved pieces of glass or plastic that bend rays of light and can magnify objects, making them appear big than they actually are. So light microscope magnifies an object in two stages. So light from the mirror is reflected up through the specimen or object to be viewed into the powerful objective lens, which produce the first magnification. The image produced by the objective lens is then magnified again by the eyepiece lens, which acts as a simple magnifying glass. So the magnified image can be seen by looking into the eyepiece lens. So the important factors in light microscopy include magnification, resolution, and contrast. So here is a light microscope. So this is the objective lens and this is the eyepiece. So magnification happens at two stages here. So light is reflected on the specimen and it passes through the objective lens and is reflected at the eyepiece over here. So the types of light microscope comprise of bright field microscopy, dark field microscopy, phase contrast microscopy, and fluorescence microscopy. So phase contrast microscopy was first described in 1934 by a Dutch physicist, Frits Zennike. So a phase contrast microscope makes it possible by utilizing two characteristics of light, diffraction and interference, to visualize specimens based on brightness differences. The principle of phase contrast microscopy is based on the wave nature of light rays and the fact that light rays can be in phase or out of phase. So in a phase contrast microscope, one set of light rays comes directly from the light sources. The other set comes from light that is reflected or diffracted from a particular structure in the specimen. So how does a phase contrast microscope work? So light that does not interact with the specimen is collected by the objective lens, passes through the faceplate ring and is retarded exactly 1/4 of the wavelength. So the phase shifted is not detectable by the eye, so the resulting image on the image plane in the microscope appears as a normal bright background. Whereas the light passing through one material or into another material of slightly different refractive index or thickness in the sample will undergo a change in phase. So this change in phase are translated into variations in brightness of the structure. So here's a representation of the phase contrast microscope. It has a faceplate and this is the diffracted light and the surrounding light. And here's the specimen. The light that is deflected by the specimen, it changes in phase and the image you see is of a different intensity based on these phase changes. So like you see here, if you look at a living cells in bright field and phase contrast microscope, this is how they would look. So in bright field they would all just look bright like this, and in phase contrast the light intensities would be different based on the thickness of the specimen of even the different cellular components of the specimen. The light microscope can be used to provide information about the activity of cells and look at very small structures such as nanostructures. And different adaptations can help to enhance images such as phase contrast microscopy, which provides contrast between cells and the solution they are in. And light microscope is a great use in pathology labs so as to identify diseases, and light microscopy can be used to explore the time and space related dynamics of molecules. Light microscopy also has a number of applications in different sectors, including gemology, metallurgy, and chemistry. But in terms of biology, it is one of the least invasive technique for looking at living cells. Phase contrast microscope is most useful in examining intracellular components of living cells at relatively high resolution. So So students in schools and colleges had benefited by the use of a light microscope for conducting their experiments, and it's widely used in microbiology and tissue culture research to detect bacteria, cellular organelles, or for testing a cell and organelle preparation for cell lysis. Now let's again see which of these techniques can be used to study the conformational changes of myoglobin. So fluorescence spectroscopy, so we saw that it's a technique that measures the emission of light by fluorophores within a molecule. And myoglobin contains the fluorophore tryptophan, which can undergo changes in fluorescence intensity and emission wavelength based on its local environment, like when it undergoes conformational changes. So, by monitoring changes in the fluorescence properties of these tryptophan residues, we can gain insights about the conformational changes in myoglobin. So, the next option was circular dichroism spectroscopy, R, like it was option R in the question. So this measures the differential absorption of left and right circularly polarized light by chiral molecules. So myoglobin has a characteristic CD spectrum that reflects its secondary structure content, including the alpha and helix and beta strands. And conformational changes in myoglobin, like the alterations in secondary structures, can be detected by changes in the CD spectrum. So these two are the answers to our question. So now let's see why mass spectrometry and light microscopy are not relevant options. So mass spectroscopy is mostly used for determining the mass and composition of molecules, but it does not give detailed information about the conformational changes in proteins. And then light microscopy is a technique for just visualizing the cellular or tissue structures, and it may not provide the level of structural detail needed to study the conformational changes at the molecular level in myoglobin. So the correct answer to the question would be option C, Q and R only, because fluorescence spectroscopy and circular dichroism spectroscopy can be used to study the conformational changes in myoglobin, but not mass spec and light microscopy. So that answers this question.